Hi, today we'll talk about the adrenal hormones. And actually the adrenal hormones comes from the, or secreted from the adrenal glands. So as you can see from the figure, adrenal glands are small triangular glands that is loosely attached by connective tissue to the kidneys. And you can see you have a pair of adrenal glands just on top of the two kidneys. And it is divided majorly into two parts. The outer, which is the adrenal cortex, and the inner, you can see the cortex, the thickness of the cortex. Let's have a pointer. So the outer cortex, this thickness, and then inner medulla, so two regions. Basically, each region is concerned with certain kinds of hormones. So if you look closely to the cortex, it's even divided into three layers: zona glomerulosa, and zona fasciculata, and zona reticularis. As you can see, there is a differentiation, and as well here is the adrenal medulla, which is the part which is inside. So majorly the cortex is divided into three regions, and you can see that the adrenal gland is covered externally by a connective tissue capsule which cover the whole adrenal gland. So the two adrenal glands are located anterior to the kidney. In orientation, it is called anterior. And they are capped or covered in a connective tissue capsule and covered in an island of fats. There is a lot of fats around it. So you can see here is the kidney, and here is the adrenal gland, and there is a fibrous tissue covering, and there is islands of fats covering the whole structure, as you can see, okay? So like the kidney, the adrenal gland lie beneath the peritoneum. What's the peritoneum? Peritoneum, actually, it's the layer which surround the whole internal organs, our organs. And the exact location relative to the kidney and the shape of the adrenal gland vary among species. So the shape and the location of the adrenal gland, as it says, varies among species. What's the functional anatomy of the adrenal gland? So mammalian adrenal gland, as we said, have two distinct regions: the inner medulla, which is majorly the source of catecholamines, majorly the two famous hormones, which is the epinephrine and norepinephrine. The principal cell type is called a chromaffin cell, and the medulla is rich in preganglionic sympathetic fibers, which actually is extension of the sympathetic nervous system. That's mean you're not able to control any impulses or stimuli to the adrenal gland. While the outer cortex is a source of another class of hormones, which is the steroid hormones, the glucocorticoids and mineral corticoids, plus a few others. The cortex, as we said, consists of three different zones, which differs majorly in the type of the hormone they secrete. As you can see here, the medulla is in the middle, and then you have the cortex of three layers: the outer glomerulosa, middle cortical region, which is the fasciculata, and the inner cortical region, which is the zona reticularis. And you can see that in the medulla, most of the hormones that secreted majorly the hormones which is secreted from the medulla is the catecholamines, which is the epinephrine, norepinephrine. While if you go to the glomerulosa, you will find that majorly the aldosterone and corticosterones, actually the corticosterones, which is the same as some people used to have cortisone tablets. So majorly that's the natural cortisone, comes from the glomerulosa. And these are the mineral corticosteroids. Mineral corticoids, while the glucocorticoids comes from the middle cortical regions, especially as well the cortisol and cortisone as well, another version of the cortisone. While the inner cortical region give rise to the androgens, which majorly the estrogens and testosterone. And you can see as well the medulla can give rise to some peptides, which act as hormones as well, which is the somatostatin and substance B. So let's focus now on each region, the adrenal first, the adrenal medulla. As we said, it's an extension of the sympathetic nervous system, so it acts as a peripheral amplifier to the signal. So it activated by some stimuli as the sympathetic nervous system, example, the exercise and cold and stress and hemorrhage, etc. All these behaviors lead to overstimulation and need a lot of the hormone responsible for the increasing the response of the individual. So the hormone synthesized in adrenal medulla, as we said, is called catecholamines. Dopamine and adrenaline and noradrenaline, which actually can be named epinephrine and norepinephrine. 80% of the released catecholamine is epinephrine. So epinephrine is a major hormone that is released from medulla and secreted and stored in the adrenal medulla and released in the circulation upon needed when the body needs. So it needs to be ready for use. So you can see here, the signal comes initially from the spinal cord. You can see that the spinal cord have the cell body of the neuron and the extension of the preganglionic, which actually the axon of the nerve, preganglionic sympathetic neuron reach to the medulla where acetylcholine is released, which is a neurotransmitter that affected the chromaffin cell. As we said, it is a major cell type of the medulla, and actually it is a modified post-ganglionic sympathetic neuron. So we have a preganglionic sympathetic neuron and post-ganglionic sympathetic neuron. Once this one is activated by the acetylcholine in the adrenal medulla, it will secrete epinephrine as a neurohormone that enters the blood and go to the target tissue. So let's see here how the catecholamine is biosynthesis. So you can see it starts with the phenylalanine. It starts with the phenylalanine. Once the phenylalanine, upon the action of the enzyme that add a hydroxyl group, it's a phenylalanine hydroxylase, and hydroxyl group is added on this position on the benzene ring. Soon after, once it is added on this position, the phenylalanine is converted to L-tyrosine. Once on the action of the tyrosine hydroxylase, another hydroxylase, which add another hydroxyl group on another carbon of the benzene ring, now the structure is called L-DOPA. Soon after that, a dopamine is formed by the action of a decarboxylase that remove the COH carboxylic group here that removes from this carbon, and now you have the dopamine. The dopamine can be converted to norepinephrine by the action of the dopamine beta hydroxylase, and you can see it adds a hydroxyl group on this position on the carbon. Soon after that, the norepinephrine is converted to epinephrine by the action of the phenylphenolamine methyltransferase by adding actually a methyl group or methyl group on the terminal side chain, and you can see here the difference. So majorly, the epinephrine, as we said, gets you ready to fight or run. So it heightens your senses, tenses your muscles, opening breathing passages, etc. in response to stress. As well, it takes less than 30 seconds to kick. So it's very fast acting hormone and lasts for several minutes. You can see here that the major difference between the norepinephrine and the epinephrine is the presence of a methyl group in the epinephrine, which is absent in the norepinephrine, and the presence of hydroxyl group in the norepinephrine, which is absent in the epinephrine. So let's see now the adrenergic receptor and the mechanism of action. So now we have the hormones coming from the adrenal medulla, and it needs a receptor to interact with and convey the signal to the target organ or target cell. So basically, there are 4 types of adrenergic receptors. The first one is the alpha-1 receptor, and majorly the effect when binding of the epinephrine to the alpha-1 receptor, you can see that they increase the blood return to the heart, increase generally the circulation, and increase the blood pressure by increasing the vasoconstriction, increase the peripheral vascular resistance, and decrease the mucosal edema. While the second receptor is alpha-2 receptor, and actually it inhibits the release of the norepinephrine through the process of decreasing the vasoconstriction and decreasing the blood pressure. So you can see that These are two different receptors, and you can see that they act against each other once the is bound by the signal, which is the epinephrine. So basically, epinephrine can do two different action depending on the kind of the receptor they interact with. The third receptor is a beta-1 receptor, and its major effect once the epinephrine binds to the beta-1 receptor is to increase the heart rate. The final receptor is a beta-2 receptor, which leads to the relaxation of the smooth muscle in the bronchi, as well as in the uterus and the peripheral blood vessel. So it leads to increase in bronchodilation, increase in vasodilation, increase in the process of glycogenolysis, and as well decrease mediator release, which is not a major effect. For dopamine, it has a specific receptor which is a dopaminergic receptor, and this leads to dilation of the vessels and increase the blood flow, and it only works with dopamine. So let's see a diagrammatic sketch to see a bit closely the effect of adrenaline noradrenaline. So you can see here the hormone. They have three major actions can do. Of course, you all know the G protein, which is a protein bound to the membrane, and actually can bound to the receptor upon activation. And upon subsequent cleavage, it leads to activation of an effector molecule. So you can see here either the adrenaline or noradrenaline, once bind to the beta receptor, it leads to the activation of the G protein that lead to the activation to the effector molecule, which is adenyl cyclase, as we said before, that convert ATP into cyclic AMP. And cyclic AMP will increase the heart muscle contraction and the smooth muscle relaxation, as well as the glycogenolysis. All these processes lead to more glucose in the blood to cope with the demand for the metabolism of the body. In this case, the type of the G protein that undergo this route of or this mechanism, we call it GS, and S stands for stimulatory. So it leads to it means that it is stimulate, overreact of the downstream signaling. While the other cascade of reaction, once the noradrenaline or the adrenaline binds to the alpha-2 receptor, here the G, you can notice that it's now called GI, and I stands for inhibitory effect. So you can see it blocks actually the signal which was activated by the GS, it blocks the signal. So it actually, when it blocks the signal, you can see that all the process that after that it will be reversed. So you can see that the smooth muscle now is being contracted, otherwise it was relaxed in case of GS. As well, it blocks the release of calcium. So majorly calcium, if you remember, it's an important mediator that help in contraction of muscles, and of course it will inhibit the transmitter release and will lead to decrease the severity of the contraction of the muscle. So you can see here how the receptor, once it differs, although it is the same hormone, can lead to a different mechanism of action, depending actually as well on the interaction of the G protein and its effect. There is a third type of G protein. Once it's bound with alpha-1 receptor, it's called GQ, and actually the gene is called GQ. The gene responsible for the production of this G protein is called GQ. And in this GQ, the story is different. It's not working on the adenyl cyclase, but it's working on the phospholipase C. And if you remember phospholipase C, from the name, it degrades the phosphoinositol diphosphate into IP3 and diacylglycerol DAG. And of course, IP3, if you remember, it leads to opening of the channels on the endoplasmic reticulum to release the calcium, which leads to smooth muscle contraction. So basically, this figure explains more the mechanism of the same hormone once bound to different receptor. So let's here summarize the response of some selected tissue to the organ and the physiological effect and the response as well, and the type of the receptor it involved in this mechanism. So for cardiovascular and respiratory effect like heart, atria, and ventricles and sinoatrial node, you can see that the receptor involved is a beta-1 receptors, and the physiological effect lead to the increase in the contractility and increase as well in the conduction velocity. That's mean the transmission of impulses. So this will lead, of course, to increase in the heart rate and the cardiac output. So beta-1 receptor is involved by increase to the cardiac output and increase heart rate. While for alpha receptor in the renal arteries and the splanchnic arteries, it leads to the vasoconstriction, which is opposed if the hormone binds to the beta receptor in the skeletal muscles, lead to vasodilation. So basically, these are two opposite processes. Of course, with vasoconstriction, it leads to decrease the blood flow, and lead. This here, it's collectively all the responses. So it may lead to decreased local blood flow, increase the pressure, of course, because when you do vasoconstriction, the pressure will be increased. And in case of the vasodilation, of course, both of these responses will be reversed. So the blood flow will be increased and the pressure will be decreased. In the veins, you can see another type of the receptor, which is alpha-2 receptors on the cells of the veins, which when bound to the hormone will lead to vasoconstriction, and of course it will increase the cardiac output and the blood return to the heart, the speed of the blood returning to the heart. You can notice that all of this adds in to increase the speed of the circulation. In the lungs, you can see that, you know, of course, there is a smooth muscles in the lungs where it have a beta-2 receptor and lead to the relaxation for the bronchodilation. So the bronchodilation will enable to have a more space for the air coming in. While the metabolic effect, you can see it in the liver, either alpha-1 or beta-2, and this will lead either to glycogenolysis or a glucogenesis, and these, both of them will lead to increase in the blood glucose. While in muscles as well, beta-2 is present, and this will as well lead to increase of the blood lactate. That means the muscles are working and lactic acid is being removed from the muscle due to the contractility of the muscles, as well as increasing of the blood glucose to provide a lot of energy source for the muscles. So that's why glucose utilization is decreased. For the pancreas, you have alpha-2 and beta-2 receptors, and of course, you can see that both of them are antagonistic, one which lead to decrease in the hormone, the secretion of the insulin hormone, and of course will increase blood glucose level, and the other one, the beta-2, is vice versa for the alpha-2. In the adipose tissue, basically it's kind of the same process which is concerned with the glucose, but in this case it deals with the lipolysis. The beta-1, 2, or beta-3, it's not proved yet, increase lipolysis, while alpha-2 decrease lipolysis. And of course, this will have their effect on the fatty acids. So basically, this slide explains which type of receptor, which type of adrenal receptors, and in which type of the tissue, and their physiological effects and responses as well. So the peripheral effects are dependent on the type and ratio of receptor, as we just said, of the adrenergic receptor in target tissue. So the relative, we can see that there is a relative effect on alpha and beta receptor. So for example, the norepinephrine, once bound to alpha receptor, it has a major effect than when bound to the beta receptor. For epinephrine, it seems that there is no difference on its effect when bound to the either alpha or beta. So here, let's take an example of how the epinephrine plasma concentration and how this is associated with its physiological response. So they measured the concentration of epinephrine in picogram per ml, and they found once the concentration is relevant to the physiological response. So normally if you are sleeping and in resting condition, you will have an epinephrine or adrenaline in the concentration of 40 picogram per ml, while when it reaches 50 to 100, this will increase a little bit the heart rate. Upon reaching a maximum of 125, this will increase the systolic pressure as well increase lipolysis. Upon reaching 200 picogram per ml, this will decrease diastolic pressure and increase liver glucose release, which is obvious because the body is needing a lot of a source of energy, which is majorly the glucose, and as well increase blood lactate due to the effect of contraction of the muscles, as well you can see as well, which is obvious as well, increasing of the keto acid synthesis. Upon reaching 400 or above 400 picogram per ml, this will lead to decrease in insulin secretion, because you know, of course, insulin decrease the blood sugar level. So in case that the adrenaline reach a 400 picogram, the insulin must disappear in order to give room for the glucose to be secreted. Otherwise, the body will sense there is a lot of glucose in the blood and the insulin will take action. And this will cancel the effect of the epinephrine or the the epinephrine or the adrenaline. So researches have been shown that vigorous exercise prevent diabetes in young mice and the humans that overeat. Okay, so it's like it's a preventive behavior. If you do vigorous exercise, this will prevent to some extent, as the studies said, diabetes in the humans that overeat, that eat a lot. Okay, so you can see here the mechanism. Actually, the epinephrine or the adrenaline, once bound to the receptor, we're talking about GS here, it will activate, as we said, the adenylyl cyclase, that soon it forms a cyclic AMP that activate PKA. So PKA now have a major role in this glucose release. So you can see here, generally it leads to short-term glucose increase. So this is the first mechanism to increase the amount of glucose in the blood. So how did PKA do this job? Actually, it does the job, just hold on. It does the job, so how it does this job? Actually, it does the job by activating phosphorylase A and inhibiting synthase B. So both of these enzymes actually is antagonistic to each other. And if you study biochemistry, you know well this mechanism. So glycogen is converted to glucose, and the other way is inhibited, leading to increase in the amount of glucose. As well, insulin, when bind to the IRS receptor, insulin receptor substrate, it induces the activation of another kinase, the kinase, PI3 kinase, that phosphorylate PIP2 into PIP3. And here PIP3 act as well as a second messenger that activate another kinase, which is BKB. BKB is very crucial in activating vesicles internalized in the cytoplasm, which is called GLUT4 vesicles. So what is a GLUT4 vesicles? Actually, it's a vesicle inside the cytoplasm that have GLUT4, which is a glucose transporter type 4. There's around 8 or 9 GLUT transporters. These GLUT transporters are being synthesized and stored in the cytoplasm in the shape of vesicles. Once the insulin is attached to the receptor, this GLUT4 vesicles is activated with BKB to be recruited to, we'll see later, to be recruited into the plasma membrane. But as well, the GLUT4 vesicles need signals coming from a kinase, which is called AMPK, adenosine monophosphate kinase, and this AMPK only activated when the ratio of the AMP over ATP increase. That's mean there is a burning out of the ATP that existing inside the cell. So this AMP, which arises from the cyclic AMP, will increase the ratio of AMP to ATP, leading to the activation of the AMP kinase that add to the signal of the BKB to activate the GLUT4 vesicles to be recruited to the plasma membrane for the release of the glucose to the blood. As well, the AMPK will activate another important protein, which is the BGC1 alpha, that promote the mitochondria to do more mitochondrial biogenesis. And of course, you all know that the mitochondria is a major source for the formation of ATP, and this will lead to the long-term glucose decrease. However, they found as well that the exercise, although we said that vigorous exercise prevents diabetes in young mice and humans that overeat, they found that exercise is less effective treatment in older type 2 human diabetes and had no effect on glucose or insulin receptor in older diabetic mice. So it seems that the exercise is only working as a preventive mechanism, okay? So let's see another elaboration on the effect of this hormone. So you can see here glucagon. If you know, we'll talk in details about glucagon in next lectures. So glucagon and noradrenaline, which is activated through the cyclic MB, once they bound to the receptor, as you can see, they activate the G protein, the major G protein, which soon will activate the adenyl cyclase. And as you know, the adenyl cyclase will convert ATP into cyclic AMP upon the action of phosphodiesterase. As we said before, it will lead to AMP. Okay, so the insulin activate this phosphodiesterase. So the cyclic AMP is hydrolyzed, as you can see, to AMP. In this event, one of the important enzymes, which is HCl hormone sensitive lipase, this hormone sensitive lipase is inactivated. So actually, what's the hormone sensitive lipase? So you can see here that the cyclic AMP will activate the kinase, as we have shown in the previous slide. It activates the BKA. So the cyclic MB. Once this active kinase exists in the cytoplasm, it will convert the inactive hormone sensitive lipase to active hormone sensitive lipase once it is phosphorylated, which lead to the conversion of the triglyceride into a free fatty acid. And that's why it's obvious that the insulin inactivate the hormone sensitive lipase. So actually this slide explains the cascade reaction that lead to the activation of the hormone sensitive lipase that lead by the end into a formation of a free fatty acids. So as well, caffeine inactivate this phosphodiesterase. So we will have no AMP in the cytoplasm. So so that's mean it remains high level, and so the lipase is activated. Again, this figure shows the effect of adrenaline on the glucose level. Nothing new in this figure. You can see the adrenaline bind the receptor, and here you can see that the G protein with its 3 subunits alpha, beta, and gamma. Once activated, it is cleaved, and the alpha subunit activate the adenyl cyclase in order to convert the ATP into cyclic AMP. Once the cyclic AMP is activated, it will activate the protein kinase A, which is inactive. It will activate it into protein kinase A active. You can see the phosphorylation signal is above here. That soon it will, this protein kinase A will activate the phosphorylase kinase, as well as the glycogen phosphorylase will be activated. And you can see here that the glycogen now is converted to glycogen 1-phosphate to be able to be converted into glucose and released through the GLUT4 receptors, GLUT4 channel or transporter to be released in the bloodstream. Just keep in mind that this protein is considered a GTP binding protein that is converted, that it can switch between two forms, the GTP and GDP. So actually, let's talk about the difference between the epinephrine and norepinephrine. Basically, or chemically, they are very similar. You remember that the difference majorly in the presence of a methyl group and the hydrogen group in place of that methyl group as well. There is a hydroxyl group in the epinephrine, in the norepinephrine. So you can see that norepinephrine is secreted as well by certain neurons in the brain, as well as the adrenal glands. So there is another input for the norepinephrine, while the epinephrine only secreted from the adrenal gland. However, the epinephrine works on both alpha and beta receptor, while the norepinephrine only works on alpha receptors. The alpha receptors, as we said before, are only found in arteries, while the beta receptors are found in heart, lungs, arteries of the skeletal muscles. It is this distinction or this difference that causes epinephrine and norepinephrine to have different function, because they bound to different receptors and as well the receptors on different target tissues. So epinephrine, the action is way higher than the norepinephrine's in terms of the cardiac stimulation, leading of course to cardiac increase in the cardiac output, and this is accounted to the beta stimulation, beta receptor stimulation, while epinephrine can have a lower effect than norepinephrine in terms of the constriction of blood vessels, and this will increase the peripheral resistance and increase as well the arterial pressure. And you can see as well that the epinephrine have a way higher norepinephrine effect in terms of the increased metabolism. Actually, it was found it's a 5 or 10 times more effective than the norepinephrine. So the epinephrine and norepinephrine are very similar neurotransmitters and hormones as well. While the epinephrine has a slightly more than an effect on heart, norepinephrine has more effect on the blood vessels, but both play a role in the body natural fight or flight response to stress. So it's like a coordinated action between both hormones. Okay, any questions? Okay. So what is the disease states in case of malfunctioning of epinephrine or norepinephrine? So what happens if there is a deficiency in epinephrine and norepinephrine? This can contribute to a lot of physical and mental conditions, include anxiety, depression, fibromyalgia, that's it's something with fibrosis with the muscles, hypoglycemia, of course, migraine headache, restless leg syndrome, sleep disorders, and some chronic stress and poor nutrition. So what happens if there is too much of the epinephrine and norepinephrine? Of course, it will be obvious to increase the high blood pressure, anxiety as well, excessive sweating because you have a lot, you have a high metabolism, heart palpitation, and as well headache. Okay, so this actually the disorder or the disease states upon the increased level and decreased level of the both hormone epinephrine and norepinephrine. So some medical condition cause people to have too much epinephrine or norepinephrine or both of them. This includes one of the major disorder is pheochromocytoma, which is tumor that forms in the adrenal gland. Actually, it's a catecholamine secreting tumors, and you know that catecholamine is secreted from the medulla of the adrenal gland. So catecholamine secreting tumors of chromaffin cells of the adrenal medulla, and in this case it's called adrenal phocochromocytoma. While the paraganglioma, it's a tumor that forms on the outside of the adrenal glands. So it is actually a catechol secreting tumor of the sympathetic paraganglia. And this, another name for that case is called extra-adrenal phocochromocytoma, because as we said, it's outside the adrenal gland. So the signs and symptoms of this disorder, actually it's treatment resistant for the hypertension, headache, sweating, palpitation, chest pain, anxiety, glucose intolerance, and increased metabolic rate. And these are all obvious symptoms of higher, because it mimics if we have increase in the epinephrine, because actually it's a tumor, so you have a lot of cells, so a lot of hormones is being secreted into the circulation that lead to all these effects. So it can be diagnosed by measuring the plasma concentration of the catecholamines, and you can see that once it's very high, higher than the normal range, and this will lead to increased as well the metabolites that exist in urine. And it can be treated by surgical resection, so just remove the adrenal gland or the part that have the tumor, and it can be substituted by drugs. So you can see here from the figure, actually the difference between the location of the adrenal one, the adrenal condition, you can see it's the red portion here, and it exists inside the adrenal gland. While the extra adrenal condition, you can see it can be scattered all over the sympathetic nervous system, which is represented by the green dots, the green lines here, and the nodes here, which represent the sympathetic nervous system. So you can see all these red batches represent the tumor that occur extra or outside the adrenal gland. And this majorly, this slide describes the difference or the location difference between the two conditions of the tumor that can arise due to defect in the adrenal gland. And this concludes our lecture for today.